Good morning. Topic for the day is Bacillus anthracis. Anthrax is a zoonotic disease caused by the spore-forming obligate aerobe Bacillus anthracis. This disease mainly affects animals, and humans acquire the infection through contact with an infected animal or inhaling spores. Historical importance of anthrax. This was the first pathogenic bacterium seen under a microscope by the scientist Polander in 1849. And this was the first bacterium to be isolated in pure culture by Robert Koch. And anthrax vaccine was the first live attenuated bacterial vaccine produced by Louis Pasteur. Now, what is this Bacillus anthracis? Bacillus anthracis is a gram-positive bacteria. They are non-acid fast, non-motile, large, rectangular, spore-forming bacilli. Spores are refractile, oval, and central in position and non-bulging. And in infected tissue, bacilli form singly or in pairs or in short chains. The entire chain is surrounded by a polypeptide capsule. And in culture, bacilli are arranged in long chains which resembles a bamboo stick. Hence, it is known as bamboo stick appearance. This is a pictorial representation of the gram staining image of Bacillus anthracis. And gram staining, clear unstained endospores. And in endospore staining, anthrax spore is used in bioterrorism. Cultural characteristics. It's a strict aerobe, and at times it may be a facultative anaerobe. Nutrient agar, the colony morphology is a medusa head appearance, that is a raised, opaque, grayish-white colony. They produce non-hemolytic colonies in blood agar. Gelatin liquefaction is a common feature shown by this organism, Bacillus anthracis, like an inverted fir-tree appearance. And the selective media used is plet medium, that is hard infusion agar with polymyxin, lysozyme, EDTA, and thalassacetate. Solid media with penicillin, that is string of pearl appearance, large spherical chains susceptible to gamma phage. This is an image showing inverted filtry appearance, that is gelatin liquefaction, and the structure purse string pearl appearance in the culture media. Biochemical reaction. Bacillus anthracis ferments glucose, maltose, and sucrose with acid, and there is no production of gas. These organisms are catalase positive and they reduce nitrate. Now, the mode of infection is mainly by the ingestion of spores present in soil, that is for animals, and in humans, inhalation of spores or the entry of spores via cut, abrasion, etc. Usually, this is a septicemic infection, and at times it shows a localized appearance. Virulence factors and pathogenesis. Pathogenesis of anthrax is mainly due to two important virulence factors, that is, anthrax toxin and capsule. Anthrax toxin is a tripartite toxin consisting mainly of three fragments, that is, an edema factor. It is the active fragment which acts as an adenyl cyclase and increased hostile CMP. This is responsible for edema. Protective factor, binding fragment that binds to the host cell receptors and facilitates the entry of other fragments into the host cell. And the last one, lethal factor, which causes the cell death and acts by cleaving host cell MAPK, that is, mitogen activated protein kinases. Toxin synthesis is controlled by a plasmid PX01, that is, they are susceptible to bacteriophage, and loss of plasmid makes the strain virulent. And the second point is an anthrax capsule. This is of polypeptide in nature and it is coded by PX02 plasmid. They inhibit complement mediated phagocytosis. Now, clinical manifestation of anthrax. First one is animal anthrax. Animal anthrax is a zoonotic disease. They often affect cattle, sheep, and less often horses, pigs, etc. The infection occurs in animals by ingestion of the spores present in soil, and they directly spread from animal to animal. And certain cases, this direct spread from animal to animal is rare. Anthrax in animal is present as a fatal septicemia or buccal cutaneous lesion produced rarely. Infected animal discharge large number of bacilli from the mouth, nose, and rectum. These bacilli sporulate in soil and remain as a source of infection for man. Human anthrax. Humans are infected from the infected animals, and in humans, three main clinical types are seen based on the route of infection. First one, cutaneous anthrax. Second one, pulmonary anthrax. Third one, intestinal anthrax. Pulmonary anthrax, also known as wool sorter's disease. Intestinal anthrax is rare by consuming improperly cooked food, and Hypotus disease or cutaneous anthrax occasionally lead to septicemia and meningitis. Cutaneous anthrax. In cutaneous anthrax, the spores enter the system via abraded skin. Face, neck, hands, and back are commonly affected. Susceptible hosts are farmers and persons handling carcasses. Incubation period of this organism is about 4 days, and the lesion start as a papule, a vesicle ring containing fluid around papule. It becomes pustule, ulcerates, and dries to become a coal black necrotic eschar. Acute inflammatory reaction leads to congestion and edema of area with a central necrotic lesion. Eschar blackens and thickens, leading to a malignant pustule. These pictures are of malignant pustule. That is first one, ulcer and vesicle ring, and second one, black eschar, but the redness remains. The picture shows the edema and the typical lesions associated with anthrax. Pulmonary anthrax. Pulmonary anthrax is due to the inhalation of dust, filaments of wool from infected animals. This is characterized by hemorrhagic pneumonia. Bacilli spread via lymphatics to blood, leads to bacteremia, hemorrhagic mediastinitis, and hemorrhagic meningitis. Onset is abrupt. Patient is acutely ill in few hours. Fever, dyspnea, cyanosis, tachycardia, feeble pulse, hypotension, vomiting, sweating, anxiety, etc. are common symptoms associated with anthrax, and death may occur within 2 or 3 days if untreated. Pulmonary anthrax, that is, spores are inhaled. Anthrax spores enter lungs and travel to alveolar spaces. And spores are transported through the lymph system to the mediastinal lymph nodes where they mix toxin that are deadly. Intestinal anthrax, which is rare, occurs due to the ingestion of spores contaminated with meat of animals dying of anthrax. Highly fatal and manifest bloody diarrhea. Now, laboratory diagnosis. Swabs, fluid, pus from pustules, sputum, blood, CSF, etc. are the common specimen used for the laboratory diagnosis of anthrax. But it has to be very much careful to take the specimen and protective measures to be adopted by the laboratory personnel. For microscopy, gram staining is used. Gram staining shows chains of large gram-positive bacilli. In dyeing preparation, capsule appears as clear halo around the bacterium. Direct immunofluorescence test detects capsular antigen. McFadden's reaction show amorphous purple capsule surrounding blue bacilli, that is polychrome methylene blue stain. Ascoli's thermoprecipitin test for the rapid diagnosis of the disease. This is done by grounding the tissues in saline, boiled for 5 minutes and filtered. This extract is overlaid on anti-anthrax serum in a narrow tube. A ring of precipitation appears at junction of two liquids within 5 minutes. This is a result of Ascoli's thermoprecipitin and the McFadden's reaction. Culture. Specimen is inoculated on nutrient agar, incubated for about 37 degrees Celsius at 24 hours, and it shows the Medusa head colonies. Gram staining, gram positive spore-bearing bacilli. Blood agar, dry, wrinkled, non-hemolytic colonies, and in selective medium blood, sodium medium with penicillin. Animal inoculation can be done with a guinea pig. Guinea pig injected with the exudate or culture. Animal dies in 36 to 48 hours. Smear from blood shows gram-positive spore-bearing bacilli. Antibody in blood is detected by ELISA, immunodiffusion in gel method, and molecular method is by PCR. Treatment and prophylaxis. Doxycycline and ciprofloxacin and clindamycin and rifampicin for about 60 days. Prophylaxis is maintaining Prophylaxis is maintaining factory hygiene, proper sterilization of animal products, etc. Immunization. Active immunization by alum precipitated toxoid vaccine and prepared from protective antigen. Three doses are given intramuscularly at intervals of about 6 weeks between each doses, given for occupationally exposed people, a live attenuated, non-capsulated spore vaccine for animals. Bacillus cereus is yet another gram-positive bacteria causing diarrhea or food poisoning. Gram-positive, motile, non-capsulated bacilli. They are the important agent for food poisoning. They are widely distributed in nature, such as soil, vegetables, milk, cereals, spices, meat, poultry, etc. Spores survive cooking and germinate into vegetative bacilli. They produce enterotoxin causing food poisoning. Two patterns of foodborne disease they produce. First one is the diarrheal type, which is associated with a wide range of food, cooked meat, vegetables, etc. Diarrhea and abdominal pain in 18 to 16 hours after ingestion of the contaminated food. Vomiting is rare. Bacillus not found in large numbers in feces. These strains produce enterotoxin resembling heat labile toxin of E. coli. And the second one is emetic type, associated exclusively with the consumption of cooked rice, usually fried rice. Illness characterized by acute nausea and vomiting, 1 to 5 hours after meal. Diarrhea, not common. These strains produce a toxin resembling staphylococcal enterotoxin. Mode of action is by the stimulation of CAMP system. Diagnosis. Diagnosis is made by analyzing the suspected food and taking feces vomitus sample, which is cultured on nutrient agar or mannitol, earth yolk, phenol, red, polymyxin agar, MYPA medium. Spore bearing gram positive bacilli are seen, and the treatment is by mild self limiting treatment, electrolytes, and no specific treatment is given. And that's all for today. Thank you.